What this is about is a technique called chromatography. I'm going to walk you through the ideas of chromatography and show you what you need to do for this week. Finally, we have a chance you guys can do this at home, a real lab, hands-on, with some basic things you can find at the store, hopefully. So I know some of this stuff might not be available because of people making hand sanitizer out of isopropyl alcohol and maybe having a lot of time to do their nails at home because all the salons are closed. And so anyway, some of this stuff might be hard to find, but if you can, that's great. And if you can't, work with your lab partner on it and see if they're able to get some of this stuff together, okay? So the basic idea is that we are taking a complex mixture and we're going to separate it based on polarity. So we've already done that in two different ways before this. We talked about crystallization. So you're using polarity and like dissolves like and different temperatures to precipitate compounds based on their solubility, which is based on polarity for the most part. Then we also talked about extraction last week. You're looking at separating things that are water soluble from things that are organic soluble. And you get two layers and things are not as gray. They're either water soluble or they're not, but in real life, some things are a little bit water soluble or some things are a little organic soluble. So you don't get 100% recovery. Now this week, we're going to talk about another way to separate things by polarity. This is really useful, especially for tracking reactions. Sometimes I can do a reaction in the lab and then just do a real quick analysis by taking a drop of that, running it on a TLC plate or a gas chromatogram. And by running it through, I see a separation of things, or maybe I just see one thing, you know, and that tells me, oh, great, this is the thing I'm looking for. And it's pure because I only see one thing here. Or maybe I can tell, oh, this reaction is not quite done because I see two spots. I see a spot of product and I see a spot for reactant. So this is a really nice visual technique that you can use in the lab for all kinds of things. It's used in forensics, it's used in food science, it's used in medicine or pharmaceutical industry. So it's a really, really useful technique. So there are two flavors of paper chromatography I'm going to walk you through. You could either run dyes like food coloring or ink or markers, whatever you have available to you. Or you can use pigments from foods. For example, at my snack today are some frozen blueberries here. And you can crush them up and you can try to extract some of the colors from different foods. And that's what experiment 16 is about, is using spinach, grinding up spinach, mixing it with acetone and hexane, and getting those chlorophylls and carotenoids out of them. Carotenoids or carotenoids, carotene, these are orange, yellowish pigments in carrots. There's also some in spinach, believe it or not. We just can't see it because of all the chlorophyll. You can try beets, cabbage, red cabbage in particular. Some of these things I'm warning you are more water soluble than organic soluble, so they may or may not work, which I think is perfect for science because that's what science is. It doesn't always work, right? Or you're trying to figure out what does work and what doesn't work. So I would say the point of this lab isn't necessarily to get good results. It's to try something as well, to do some real science, okay? So whatever you get, please explain what you did, and if it didn't work, you can always just explain why you think it didn't work, what might you try next time that might work. And that to me is also very real science. And hopefully maybe you and your partner can work together and say, okay, I'm going to do spinach and my partner's going to do carrots, and we're going to see what the difference is. Or maybe neither of them work. Hopefully one of them will work. But again, there are so many iterations, so many combinations of variations you can do on this experiment, and some of them are great and some of them may not work. So I would just suggest that the vegetable stuff and fruits might be a little trickier to get good results, but could be really fun, okay? And fun to try to figure out what can work. All right, I'm going to share with you this more foolproof food coloring inks and markers, which did work. And again, one of you is lab partners. One of you could do the one that is very straightforward, and one of you could try the one that's a little crazier. Okay, it's really up to you. Yeah, markers will work great, and I'm going to actually share that with you right now. So I tried this last week just to see how well it would work with the things that I already had at home, so just that I wouldn't even have to go to the store. The main purpose of chromatography, and we're doing paper because that's something we can find at home, hopefully, is we're going to separate mixtures like, you know, here's a marker. It's a blue marker. It says so, right? It's a color blue. But within this blue marker, there are actually lots of other pigments. It's not just one pigment that's blue. It could be a mixture of different kinds of pigments, right? So the idea is that in markers or ink, there are a variety of compounds that make up the ink. So one thing that you can do is to separate those components. So some of the terms I'm going to walk you through are mobile phase, stationary phase, and retention factor. So we'll talk about these in the course of the experiment. And I'm going to show you some visuals of what you can do at home, okay? So just the really simple experiment is take a coffee filter. So I have this like pour over coffee system that I use at home. It's just a container with a, you know, a filter, and then I with a funnel, and I put the filter inside. And so I just had this conical filter. So I basically just cut the seam. It made 2 pieces, and then I used one to load up my samples, which are different kinds of markers. And I showed them on the right side. I used a black Sharpie, a green Sharpie, a blue Crayola marker, a purple ink pen, and a blue dry erase marker. That's probably why these are on my desk right now. Then I did the same thing over here. Now, notice that you want to use pencil to mark your paper. Do you guys know why I don't want to use a pen? Or maybe why I don't want to use one of these markers? The reason why is let's take a look here. Yeah, the ink will bleed, and that's what we're trying to get the samples to do. We want the samples to run up the filter and separate based on different speeds that they run up the paper. And if we use an ink or something like that, we're basically going to contaminate our samples. Okay, so usually we mark a starting line that we're going to use later, and then we load up our samples on there. And then we usually have little lane markers, so I just numbered them 1 through 5. And then the idea is that this paper is acting as the stationary phase. It is the thing that stays still, and the sample gets loaded on it. Now that can be many different kinds of materials. In this case, it's paper, but it could also be, in the lab, we use like a plastic sheet that has like a silica coating or an alumina coating, which is silicon oxide or aluminum oxide coating. And that acts as an absorbent material as well that the compounds can run up. So you have a variety of polarity backings on what we call a thin layer chromatography or chromatograph. It's basically a thin layer of chemical that the compounds will absorb onto and move on. Okay, so that's called thin layer chromatography, or TLC. And you'll see that in the lab experiment, TLC. We're using paper though, that's a lot more available to us. Okay, after you load up your sample and you're clear on what is what came from what, then I decided to run 2 solvent systems. One is 70% isopropyl alcohol, which is also called rubbing alcohol. And then on the right, I just have plain tap water, okay? And actually I had a lot of leftovers, so I did not have 2 glass Tupperwares. I was using all my Tupperwares that day, so I had to find this plastic one. You do have to be careful with using plastic, just because organic solvents like alcohols or acetone, you know, they can actually leach some of the plastic into the solution. Not that that's a big problem for this experiment, but just FYI, usually don't want to put organic solvents into plastic. Okay, now I did a quick time-lapse video of putting each of those identical or nearly identical filter papers into each of these solvents just to compare how different they would be. Now we call the liquid solvent, we call that the mobile phase. I think you can see how it's moving up the plate. And as it does that, I say plate, but I mean paper in this case. So as the liquid runs up and the mobile phase catches these samples, it's then moving components of the sample up through that stationary phase at different speeds. Some of you guys know this concept already, so I apologize. I think I'm assuming that just in case you've never seen this before. Basically what's happening is the solvent has a certain polarity. As it moves up the plate paper by capillary action, the way a tree draws up water, as it moves up through capillary action, the sample or the marker and inks that I have, they have a certain attraction for the stationary phase or the paper that they're sitting on, and that mobile phase, which is moving. Now, a really great analogy I heard from someone teaching chemistry was that the paper is like you walking in an airport, and that the mobile phase is like a conveyor belt. Like sometimes in the airport you have like a nice little conveyor belt that you can step on to take you through the terminal. And so let's picture that when you drop this paper into the solvent, it's like you're stepping onto the conveyor belt. And as you're moving on the conveyor belt, that's like the sample being taken up by that solvent, they have different attractions to either staying on the conveyor belt or that mobile phase, so either the alcohol or the water. Some compounds really like the alcohol, and they'll move up with the alcohol really fast. Other compounds don't like the alcohol as much, and they're not going to move so fast, okay? Same with the water. Some compounds might really like the water and move up higher with the water than if there was something else, right? So, and that's based on polarity. Like we know water is very polar. We know isopropyl alcohol has carbon groups on it, 3 carbons to be specific. And so the carbon and hydrogens make this a little more nonpolar. So it's that concept of like dissolves like again. My more nonpolar compounds really love this conveyor belt of isopropyl alcohol as a mobile phase. And so you can see that my lane 4 is really interesting. This is just a regular purple pen. I don't have it on my desk, but it's just a regular ballpoint purple ink pen. And you can see within that purple ink, I can see purples and I can see pinks. Okay, so now if I run the same ink, but with a water system as the mobile phase, look at the difference in colors. I can actually start seeing blues now. Isn't that cool? So I can see different colors being drawn up by different solvents because maybe this blue has a different polarity than this magenta, and maybe this magenta has a different polarity than whatever this is, okay? And I can see a little bit of blue in here also. So that blue is not as happy in this isopropyl alcohol as this red stuff is. So what does this tell you guys about the polarity of this pink pigment compared to this purple and blue pigment? Would you say it's more polar or non-polar relative to the stuff down here? This is isopropyl alcohol. Can you draw any conclusions about that? Or likewise, what could we say about this blue pigment relative to this pink pigment? Let's take a look here. Yeah, okay. Rasmina says more polar. Which one's more polar, Rasmina? The pink one or the purple one? Okay, the pink one. And then what can we say over here with the blue one versus the pink over here? When we run it in water, we see a difference. Yeah. Yeah, so I, maybe my first question's a little ambiguous, like, because what am I comparing it to, right? So like I have pink and purple. The pink runs up more with isopropyl alcohol. So clearly it likes isopropyl alcohol. But would it like water more? Well, when you look on the right, we see that it is moving with water, so it is somewhat polar, but we see this blue pigment all of a sudden catching up and moving past the pink pigment. So to me, that tells me the blue pigment moves with water, doesn't move with isopropanol. And we know water's more polar than isopropyl alcohol. So at least this is very clear to me that the blue pigment is polar. You can see on the left too, oh, Avery, do you mean this blue over here, or do you see over here? There's a little bit of blue here. Yeah, you can see a little bit of blue moving with isopropanol, but the blue is not moving as fast in isopropanol as it is in water. So to me, that tells me we can draw the conclusion that the blue pigment is more polar than the pink pigment. What do you guys think about comparing the green pigment? Take a look at the green pigment, my green Sharpie, which I do have on my desk. It's moving in isopropanol, but it's not moving so much in water. What does that tell you generally about the polarity of a Sharpie, green Sharpie? Does that tell you the pigment is sort of more on the non-polar or the polar side? Yeah, right. It like dissolves like. The green pigment is really moving fast with isopropanol, and it's not moving at all with water. And then the last thing I wanted to point out was, and similarly with this black Sharpie too, which makes sense, green Sharpie, black Sharpie. And unfortunately, I wasn't able to observe any colors in my particular experiment. Maybe if I tried a different alcohol or a different paper, I might actually see different colors there. And I will tell you, I have seen different colors from black Sharpie, just I can't see it in this particular example. Let's take a look at blue dry erase marker. I'm sure you guys are well familiar with these from school. It's probably where I got this pen from. So if we have a dry erase marker that's blue, there's a little bit of streaks here. So maybe it wants to move a little bit. And here, not at all. So maybe we could tell also the dry erase markers. What do you guys think? Polar, non-polar? I might want to try another solvent with this because it seems to be even more nonpolar than all the other ones. At least these moved a bit with the isopropanol. Some of them didn't move at all with water. So I need to keep going nonpolar, and maybe I would try some fingernail polish remover, either in the form of acetone or ethyl acetate, tend to be things that are more nonpolar. Okay, so I could try a third experiment and give that a try, see if anything happens there. So the cool thing about it is that by using different polarities and comparing, you can make some conclusions about the relative polarity of the various colors. And even you can see things that maybe you didn't see in one experiment, you can see these colors all of a sudden appearing in another experiment. So that should be really fun to try some different things at home and see if you can get separation like this to occur. So when you remove these from your solutions, you want to use a pencil again, and you want to use a ruler, and you want to compare where you started the sample from where it ended. And we call this ending point a solvent front. The solvent front is where it went up to. I think they use the term the front in war zones and things like that, right? You get up to the front. So normally, of course, things usually turn out better when you wait. If you wait till this thing goes higher up the paper, you will get more separation as it goes. And that's generally a really good thing. In fact, when we do something called gas or liquid chromatography, we're actually using gases and liquids as samples. sometimes we use tubing to have a stationary phase packed inside a tube, and that tube is coiled many meters long. In fact, I remember in graduate school, we had a stationary phase in a tube that one of us had to go up to the 5th floor of a building and have the tube go all the way down to the 1st floor. And we used to put the stationary phase inside that tubing, and then we would take it and we would coil it up. And so the longer you give it to separate, the better the separation will be. So, if I let this thing go much longer, maybe I would see the pink at the top and the purple at the bottom. And so you would get more separation. In this case, I think it just stopped and I couldn't get it to go any higher. So I just took it out. I marked the front, and we will use that line to do a calculation. And the calculation is, once it dries, by the way, some pigments are sensitive to oxidation by air. So if you are doing things like spinach or carrots or other fruits or vegetables, notice that those molecules can oxidize and change colors. So maybe you might be able to see some green from chlorophyll. I would recommend right away taking a picture of it. So mark it, put a ruler, and take a picture because over time, the colors will change, or you might not even be able to see them anymore. All right, so what I did was I took a picture, and I'm going to do a calculation called a retention factor, or RF. F is in retention factor, or ratio to front. So what I would do is I would sort of try to figure out how polar or non-polar these materials are. I would come up with a number to quantify how much they traveled on this plate. So that's why I have the ruler here, and they're basically just 2 measurements you need to take. Let's use this as an example. Okay, so I want to know what's the RF value for the red pigment and what's the RF value for the blue pigment. So what I would generally recommend is that I will count the red as being up to here. Can you guys see my mouse okay? I hope you can see the little cursor and where I'm pointing to. Whoa, it disappeared? Okay, I hope that, okay, great. Okay, thanks, Avery. So right here at the tip of the red, I would note what distance that traveled up. So let's say that that's about 3.5 centimeters based on the placement of my ruler. And again, you want to make sure the ruler is somewhat straight and starts at 0 on the starting line. Okay, so this is about 3.5. Then I will measure where the front is. It's about 6.7 maybe, something like that. So I would take 3.4 centimeters divided by 6.7 centimeters. I'm going to get something that's less than 1, right? It has to be on the front in order to be 1. So you're going to get fractions for all of these values, okay? So I would put RF, red pigment, and then I would calculate that ratio, whatever I said, I forgot already, 3.6 or whatever, divided by 6.7. And then I would put the RF value for the blue pigment is, okay, I would see how far that went up. I would say 4.8-ish or 5.0 divided by 6.7. So you always divide by the front distance that the solvent traveled. I'm going to write this down, don't worry, okay? So once you do that, then that sort of gives you a quantitative way of communicating to someone else, you know, let's say without drawing it out or showing them a picture. You could just say, my sample separated into 2 components, each with an RF value of blah, blah. And then you tell them what your solvent system was, and then they would get a sense for what to look for in their experiment or how to draw some kind of conclusion from that. Okay, I think the kinds of questions you want to generally ask is, you know, which samples were you able to succeed in separating a complex mixture? For example, the blue Sharpie or the blue dry erase marker, remember, it didn't move in either solvent. So I really couldn't tell you whether this was made out of a mixture because I couldn't separate anything. But plenty of other samples I was able to separate to some extent. Which samples appear more complex than others? So I just talked about that. Which samples did not work and why not? So again, with the blue dry erase marker, they didn't move in either solvent because maybe those aren't the right polarity. So maybe we need to actually go toward more non-polar solvents to see something here. And again, that's where I would suggest using acetone or something else that you might have at home. By the way, be really careful with using these experiments at home. You always want to be careful not to have little kids around who might drink the liquid, or pets, or, I mean, they can usually smell it, but one of my colleagues, actually, she had antifreeze in her garage, and her cat actually drank it, or, you know, got poisoned by it. So you just have to really be careful when you're doing stuff like this at home, even though it's, you know, all household stuff. Okay, what conclusions can you draw about the polarity of the different pigments? So it's not going to necessarily be clear if you only do one trial. Again, isopropyl alcohol, it's intermediate polarity, right? It's definitely more polar than water, which is why I chose to compare in water what was happening. But it's also more polar than other things like acetone, ethyl acetate, which is a nail polish remover. And those are more polar than gasoline or hexane or paint thinner, right? So there's a whole spectrum of polarity of things at home. And it's hard to draw the conclusion from just looking at using one solvent system. But if you only have one solvent system, consider doing a comparison with water at least, because I know you have water. If you have multiple things, like I said, you could try water versus acetone versus isopropyl alcohol, or even versus vodka, right? Vodka would be a mixture of ethanol and water. Try whatever you have at home or whatever you're interested in. All right, so that is the essence of what I wanted to talk about. Let me see if I can actually share my iPad with you guys and draw some stuff out. Before I do, though, I did want to show you on ChemDraw. I almost forgot. ChemDraw has some really cool templates in here. So if you go to this menu on the left, there's a little stamp icon for templates. There's a lot of really fun templates in here. You can go to, gosh, I forgot how. Oh no, right above templates, there's a TLC, a thin layer chromatography tool. Okay, so you can basically put in however many samples you have, and then you can move them around. And look, as you move them, you can see the RF value getting calculated as you go. So, I mean, you know, actual scientists use this as a tool. When they're publishing a paper, they might just, you know, include a little graphic of this. So, of course, they're assuming like one spot per lane. Maybe there's a way to move things over and have multiple spots also. Yeah, there we go. So anyway, you can play around with that a little bit. And so you could see, you know, this spot was at, you know, whatever RF value. And as you move it up, you see the value gets larger and closer to 1. Okay, so you could play around with that. I don't, yeah, if you double click, I'm just learning some of this stuff myself. I don't use it that much for TLC, but there might be ways to insert extra dots in this column. I don't know. I'd say you guys probably insert the number of lanes present. Let's just see what happens. Okay, so that just, yeah. Okay, never mind. I think I wanted multiple spots in one lane, but okay. Back to the assignment. There's a teacher that posted some thoughts on doing the ink chromatography, Jansen Tan. So you can look at his video if you want to see how he did it. He also walks through some what if you did this or what if you did that scenarios, just to get you to think about that. I listed some different kinds of solvents you could find at home or at a drugstore or at a grocery store. You can try the spinach, carrots, red cabbage. If you do that, I recommend you grind it up, it may be in a blender with some water, and then try to concentrate the liquid. Try not to have any solid bits. I'll try to just limit it to your liquids, and then try to figure out a way to get that to make a nice, small, compact spot on your paper. So it's a little tricky, but maybe like a dropper or something to do that with, just carefully using a spoon or a straw or something even.